Okay, chemical environment alters the pKa. So the teacher kind of likes this idea, but we're going to look at this carboxyl group. It normally has a pKa of 4.8, but if you throw on an amine group, it changes the pKa. And so when I say chemical environment alters the pKa, I mean that if you add certain groups to the carbon, it can change the pKa of that proton. And the same thing for the amine group over here. Okay, and so now we're looking at an ionizable amino acid. And you're going to be asked certain questions about ionizable amino acids. And certain questions like what is the charge um, on this amino acid or on this protein. And you're going to have done at least a couple things in order to answer these questions. The first thing is you need to have the pKr memorized. Okay, you need to have those 7 pKrs memorized. The second thing is you need to memorize if the R group has a carboxyl group or if it has a nitrogen group. For example, let's look back at some of the amino acids. So for arginine, you have to know that there's a nitrogen group. Okay. Let's see. For aspartate and glutamate, you need to know that they have carboxyl groups on their R groups. So let's go back. Okay. And so, okay. And so, earlier in this slide, I talked about carboxyl groups on the actual amino acid. And now we're going to be talking about the carboxyl group on the R group. And so remember, on certain R groups we have carboxyl groups, on certain R groups we have amine groups. And so if an R group is a carboxylic acid, if it's COOH, then the pKr in this case is 4.25. Okay, so glutamate is 4.25. That means that below a pH of 4.25, he's going to be protonated, and above a pH of 4.25, he's going to develop a charge. He's going to have lost his proton. If the R group is NH3 plus, then we always write NH3 plus on the left. We always start with NH3 plus. We draw our arrow, and then on the other side we draw NH2. And if the pKr, let's say we're talking about histidine, so the pKr would be 6, then anything below a pH of 6, you'd have that charge. Anything above a pH of 6, you would have lost your charge. So let's look at glutamate. So we would expect that below a pH of 4.25, the R group will have its proton. Anything above a pH of 4.25, you will have lost that proton. And we're just looking at the R group. And remember, if we were talking about the carboxylic acid on the coming off the central carbon, then remember his pK1 would be around 2, it's 2.34. But here we're just focusing on the R group, and so let's look at histidine. And so when we see an ionizable R group, the first thing we do is we know its pKr because we've memorized that, and then we've memorized if the R group is a COOH or an NH3. So for example, histidine, we've memorized the pKr, we've memorized that the R group has a nitrogen group on it, and so the first thing we do is we draw NH3 plus on the left, we draw an arrow to NH2, and over the top we draw 6.0. And so anything below 6.0, we would expect to have that positive charge on our R group. If we move above 6.0, then we expect to have lost that charge. Now, why is it so important to memorize if the R group is a nitrogen group or a carboxyl group? Well, let's say for histidine you accidentally thought this was a carboxyl group. Well, remember, on a carboxyl group, we always start off with COOH, which is a neutral charge. And so we would have said that the R group was neutral. And so it totally throws off your charge. Alright, so now we're going to move on to something else. This is an amino acid plus an amino acid. And so when we join amino acids together, moving down this way, that's called a condensation reaction to bring together. If we want to break that with water, then it's called hydrolysis. Alright, so just to review, we have 13 amino acids that have no ionizable R group, and so they have 2 pKa's. And the pi is an average of those 2 pKa's. There's 7 amino acids with ionizable R groups, and so they have 3 pKa's, which is pK1, pKr, and pK2, and the pi is an average of 2 of those 3 pKa's. And I'm going to have a video that shows you how to determine the pi on the ionizable R groups. And just a reminder that pi is pK1 plus pK2 divided by 2. That would work for these 13 amino acids. It's really easy to find the pi, but it's a little bit trickier for the 7 ionizable R groups, because do you use pK1 plus pK2, or do you use pK1 plus pKr, or do you use pK2 plus pKr? Alright, on a protein, we're joining amino acids, and it's a CCN repeat. And so we have CCN, CCN, CCN. If we want to make well, I should define what this means. Peptide bond is when you take one amino acid plus a second amino acid. And so if you want to make a peptide bond, that's a condensation. And remember, if you want to break, that's hydrolysis. You have the amino terminal end, which means that's the end of the protein where you have an amine group, and then a carboxyl terminal end where you have a carboxyl group. Just by looking at this carboxyl group, you can tell what the pH, at least to a certain point, what the pH has to be. And so if the carboxyl group has lost its proton, then we know the pH has to be at least above 2.3. So let's go back and just review that concept. And so remember, you just draw the COOH going to COO minus, and if the pH is above 2.35, then you have a negative charge. Alright, this is going to be an important concept. One amino acid equals 110 Daltons, which is kind of like a unit of measure or weight. The exception to that would be if you have an amino acid with lots of small amino acids or metal cofactors. Okay, so how much does cytochrome C weigh? So there's 104 residues, so you'd multiply that by 110. Residue means amino acid. Okay, just a couple definitions. A simple protein means just amino acids. A conjugated protein means amino acid plus a prosthetic group, or plus something else. And so here's a whole bunch of different types of conjugated proteins. And so this is the name of the conjugated protein. These are the names of the prosthetic groups. I wouldn't worry about memorizing any of this. Okay, we're moving on to separation techniques. We're going to use chromatography, and there's 3 types: ion exchange, size exclusion, and affinity. Okay, we're going to look at ion exchange first of all. And so we have this long tube, and we fill the tube with these beads, and those are called a matrix. And so you call the beads a matrix. And so there's 2 types of ion exchange. There's cation exchange and anion exchange. And if it's cation exchange, the matrix is full of negative beads. And so let's say you've got this beaker full of proteins, but you just want, it's full of like 10 proteins, but you just want to isolate one of those proteins. I should say this beaker is full of 10 different proteins. And so one of them might be an I protein, one of them might be a hair protein, one of them might be an albumin, But let's say you just want the albumin. And so you would maybe use ion exchange chromatography. And so these beads are negatively charged, and so anything that's negative is going to be repelled, and that's going to be uh, what's going to come out first. And so what's going to be most important about these concepts is to know what comes out first. Another word for that is elute, what elutes first. And so what's going to elute first in cation exchange? It's going to be whatever is the opposite of the name, anion. What's going to elute first in the anion exchange? It's going to be the cation. Size exclusion. What's going to elute first? Whatever's big is going to elute first, and whatever's small is going to elute last. So be careful, because this is the opposite of gel electrophoresis, where the smallest goes to the bottom first. Okay, then you have affinity, and what elutes first? Everything else, and what elutes last? Your protein. So you've got a beaker full of tons of different proteins. Let's say you just want one of those proteins, and so you know the ligand for that protein, and so you would attach those ligands to the matrix, and when you pour the proteins, everything else is going to elute first, but your protein is going to bind to that ligand. And then you use a solution saturated with your ligand, and you just pour it down there. And these ligands right here will compete with the binding sites for these proteins, and they'll kind of just like steal those proteins off of the matrix so that they can run through and elute. Okay, electrophoresis. We have a problem in that proteins have lots of different charges, and they're globular. The solution to this is SDS. Okay, and so with electrophoresis, we have a negative and a positive charge at the bottom, and the proteins migrate down based upon their size. And so if it's smaller, it's going to move down to the bottom. If it's larger, it's going to stay at the top. Sodium dodecyl sulfate is often used with electrophoresis. You want to know both names, so recognize it as SDS, just in case in the test the teacher says SDS, you'll know what he means, or if he throws this big word out, you'll know it's the same thing. And it has a negative charge to it, and SDS is going to coat the protein in negative charge, and so that the charge to size ratio is similar among all proteins now. And then it's going to denature proteins so they're no longer globular, so they're going to be straight. And so now you don't have to worry about all these proteins having different charges, because now they have a similar charge, and they're no longer globular, and so they'll all migrate at the same rate. Okay, here's a slide showing what it looks like in the lab. Okay, isoelectric focusing. This is used to find the pI. And so you have the protein, and it's going to migrate until it reaches its pI. And remember, when the pH equals the pI, that means you have a net charge of 0 on your protein. And so if you have you have a kind of like a gradient of charge going up this solution, and so it's kind of like negative at the top, kind of positive. Well, if you have a 0 charge, you're not going to be attracted one way or the other. You're going to stop. And so when that protein stops, you know that's the pI, because it has no charge. It's not going to be attracted one way or the other. And for all of these proteins, for example, we've got a known pI, and those pIs are known because of isoelectric focusing. 2D electrophoresis is used to determine molecular weight. And so the first dimension is isoelectric focusing. The second dimension is we take that beaker and we stick it on a gel, and we let those migrate down to find their molecular weight. Okay, in new terms, we've got activity and